welcome back to the lecture series in NPTEL on bioelectricity. So, we are on the 34th lecture. So, last lecture we talked about photosynthesis. So, now we will be talking about, we finished the photosynthesis, now we will be talking about the inspiration we have drawn from photosynthesis, and we will be talking about this topic at two levels. One, the inspiration which has led to the development of Gretzel cells or dye sensitized solar cells, and the second one which we will be talking about is the inspiration drawn from the manganese cluster. Just a brief recap for those of you who are joining in this lecture. So, talking about photosynthesis in the last lecture or last two lectures, we talked about So, there are two major things what we observed. One thing we observed that chlorophyll dye has the ability to absorb light and ejects an electron. So, essentially this chlorophyll dye is a natural photovoltaic material. Okay. So, this is one thing what has inspired generations, since we know this thing. The second important thing about chlorophyll in terms of the inspiration is it has the ability to split water, which is essentially, if you remember my last lecture I told you, the most currently in this current earth ecosystem, most abundant source of electrons. Okay. And this water splitting cluster which we talked about is present on just underneath PS2 or photosystem 2 or P68T. So, we want to recollect it is a manganese cluster calcium surrounded by a series of proteins which is called the oxygen evolving complex. OEC. Okay. So, these are the two zones. So, if you look at the slide, these are the two zones where we have drawn inspiration: synthetic chlorophyll as solar cells, it is this part, and the second one mimicking water splitting cluster for generation of hydrogen. There is significant amount of hydrogen which is generated out here, and this is what we call as hydrogen as fuel, where you see all these hybrids and hybrid cars and everything. Okay. These are the two things. So, talking about the chlorophyll dye, there are several other dye, and we will have an exclusive one lecture on this one, which will fall under dye sensitized solar cells. We will talk about this in detail, which is also commonly called as, after the name of its innovator, Gretzel, we call Gretzel cells. So, we will have a totally separate one lecture on Gretzel cell or dye sensitized solar cell, what are the different dyes, and in that you will come across several dyes, ruthenium dyes, and you will talk about natural flower dyes, likewise, so and so forth. Okay. Today we will be talking about hydrogen as a fuel by mimicking the manganese cluster, what are, or if not mimicking, by deriving inspiration from the manganese cluster, which is present in the photosystem 2 of the thylakoid membrane. Okay. So, let us move on what we meant by the artificial photosynthesis for solar water splitting. So, essentially hydrogen is generated from the solar driven water splitting, has the potential to be a clean, sustainable and abundant energy source. Inspired by natural photosynthesis, artificial solar splitting complex or devices are now being designed and tested. Recent development based on molecular and nanostructure designs have led to advancement in our understanding of light induced charge separation and subsequent catalytic water oxidation and reduction reaction. There is a very nice review which I have cited here in the Nature Photonics, came in the year 2012. Please go through this review, and most of the presentation which I will be doing today is from that specific paper, and there are several good references, and you will see the reference of Gretzel, wonderful paper. There is a paper by Gretzel published in Nature. The title of the paper is Photoelectrochemical Cell. Please go through this paper. There is a cross reference of this paper in this particular paper, what I am referring now. Go through this paper. This will help you to appreciate this whole thing far better. Than there is a wonderful review, wonderfully good paper in that by Helmut Trebuch. You will again see the reference in this Nature Photonics paper. Go through the work of Helmut Trebuch. So, these all will help you to get a very global idea how the world is now thinking, where it is all moving. Okay. So, coming to the solar to chemical energy conversion. Solar to chemical energy conversion is the ultimate goal for the scientist in the field of energy generation. This process does not emit any greenhouse gases, and the chemical energy can be stored and used when required, which is in the form of hydrogen. Plants form the conversion through natural photosynthesis, which is abbreviated as NPS, in which oxygen and carbohydrates are produced from water and carbon dioxide using sunlight, that we have already discussed. Okay. The energy conversion efficiency of NPS can reach around 7% under optimum conditions, although an efficiency of less than 1% is usually expected for agricultural crop over their entire life cycle. So, in the previous lecture, while I was talking to you about the efficiency of natural photosystem. So, if you remember, I was telling you that you are compromising on efficiency of natural photosynthetic systems, which is approximately 7%. The reason possibly with what I cited is, in those thylakoid complexes, there are a lot of, when the chlorophylls are getting excited and they are getting oxidized and they are ejecting an electron, there are a lot of free radicals which are formed. Now, in order to prevent free radical damage, this process is slowed down, and that is what you see, the 7% efficiency is in order to ensure that the free radicals which are formed there does not damage the system, so that we fail to recuperate the system and it ages very faster. So, possibly that could be one of the possible reasons. Okay. Now, moving on to the next slide, see the artificial photosynthesis, what we really meant by that. So, you have two words which are coming for you now. One is natural photosynthetic system and the other one is APS, which is A stands for artificial, N stands for the natural. Okay. So, coming back to the slide, a potentially more controlled technology for the solar to energy conversion, chemical energy conversion process is artificial photosynthesis, which aims to emulate natural photosynthesis using man-made materials. APS has been a fascinating scientist in field ranging from material science to physical to inorganic chemistry. However, it remains a significant challenge to construct an efficient APS device capable of producing molecular fuels such as hydrogen at a scale and cost that can compete with fossil fuels. Significant advances in efficiency are required before such devices will be able to compete with conventional energy sources. So, this is what is competing with the other fuels, like coal, petrol, diesel, and of course, you have the nuclear. So, this process has to be cheap, green, and fast scalable. Without this cannot really be achieved the way we are dreaming of a world of clean and green energy. And nature has already done it. Nature is already doing electron donation, and if you remember in the last thing I told you that if I have to put 3 words for the whole evolution, to who is the electron donor, who is the electron acceptor, and third one, who is the abundant, very abundant source of electron. That is it. This is all about this, how you really can manipulate this to the next level. Coming back to the slide. So, what are the basis of the basic of the natural photosynthesis? So, photosynthetic reactions are determined primarily by 3 reactions if you look at it. There is a light harvesting process, if you remember it, there is a chlorophyll P680, P700, which is essentially P photosystem 2 and photosystem 1 respectively, which is supported by a series of molecules which is funneling the light and ejecting the electron. Okay, this is the first foremost thing. So, you need a light harvesting, because light energy is evident. So, if you come here, who is the donor of electron? So, we are talking about what is the energy which will help you to eject the electron. If we know the donor, then that donor needs an energy which could. So, this energy is provided by the sun, and this energy is funneled through the antennae pigment to the light harvesting process. Okay. Second thing, if you go through this slide, charge generation and separation process. So, what is the charge generation we talked about? Talk about whenever there is a chlorophyll molecule sitting here, it ejects an electron and it becomes oxidized. So, this is the whole charge generation process, and the combination electron hole pairing, okay, and the separation. So, you have to have a way by which you can separate these processes. Otherwise, what will happen? They will recombine back. So, the chlorophyll which is formed, the electron is close to it. So, this is the problem. So, this chlorophyll which is there, one second, which is devoid of an electron, plus the electron which is rejected, it will recombine again, then and there. Okay, there will be a recombination process, and it will bring it back to its ground state. It will reduce it. So, there has to be a way by which you can separate the charges. You have to separate them, otherwise they will recombine. Okay. Then comes the catalytic process, which essentially is the splitting of the water. Okay. Now, the overall efficiency is determined by the balance of the thermodynamics and the kinetics of all these processes. In recent decades, intensive studies have been focused on further investigating the mechanism involved in NPS. In particular, researchers recently revealed the structure of oxygen evolving complex site in photosystem 2. thus providing new inspiration for design of APS structures. So, I was talking to you about oxygen evolving complex OEC underneath P680 or photosystem 2. So, recently, at least 3 years back, this structure has been revealed. So, now since this structure is known, there is a lot of inspiration to design better oxygen evolving complex or better water splitting complexes. Okay. Now, moving on to the next slide, NPS to APS, what are the recent development? Overall, the past decade, fundamental progress has been made in developing novel material structures for water splitting reactions, particularly in those that target an efficient oxygen evolving catalyst for use in APS devices. Nanomaterial composition and structures, including inorganic, molecular and hybrid organic inorganic material, have been explored to meet the specific requirements such as light absorbing wavelength modification, photo induced charge separation and faster water splitting reaction. Now, let us have a glimpse of diagrammatically what is happening, what are the difference between NPS and APS. The upper picture you see is the NPS, where you see the P680, which is just on top of the water splitting cluster, which is the manganese cluster on the right hand side it is shown. Now, P680 electron gets excited, that electron through pheophytin QA to QB to plastoquinone to iron sulfur to cytochrome BF to plastocyanin moves on. It is hopping down, 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 down. If you look at it in the Z scheme of things, it moves to P700, where already there is another chlorophyll molecule which has oxidized. So, that chlorophyll molecule which has oxidized has to be brought back to its ground state. So, at P700, it is this electron which brings back that P700 excited molecule back to its ground state, and the electron which is ejected by P700 then moves to AO to A1 complex to FX and FA big complex, and eventually it will works with NADPH reductase to make NADPH molecules. Okay. And in the meantime, on the extreme left, if you see, the manganese cluster is splitting the water into protons, and oxygen is a byproduct, and it is supplying the electron, the rich source of electron, what I was kind of highlighting on the board. Now, if you look at the B and C are essentially these molecules. They are the dyes which have been mimicked, okay? The chromophore or the dye or semiconductor, which upon absorbing light gets excited and donates an electron. This electron is being accepted by protons. So, you have to supply proton. If you see the B, you have to supply protons. Those protons forms hydrogen gas, whereas you need an electron donor. So, in the both cases, electron donor is 2H2O. So, electron donor is the water is splitting and forming protons. These protons are filtered. So, basically there are two reactions which are happening here. So, you have this water splitting cluster which is generating protons and oxygen. These protons are filtered through a proton membrane and added with the electron to form hydrogen gas. Okay. And on the other hand, this oxygen is a byproduct of this whole process when this light reaction is taking place. Whereas, you can do the same thing by mimicking or reducing the efficiency of the system by having two different dyes, the chromophore 1 and chromophore 2, and exactly follow the mechanism, what the Z scheme of things which is being followed by the natural photosystem. So, this is the overall A, and if you kind of go through the caption of this figure, which has been derived from that Nature Photonics paper. So, this is exactly what it will tell you, that and a two-step Z scheme of reactions of the P is the chromophore, it is the last two lines of a single step reaction, P excited state. So, that is pretty much, you can go through this, and that will give you an idea that how these photosystems are working. Now, if you have to emulate these kind of photosystems, now next I am coming to the structural designs of APS reaction processes. So, what you need essentially is, if you see the first picture, which is the A and the B. So, we will talk about the structure of the carotenoid, porphyrin and fullerene molecular diet system, which is basically this one, carotenoid, porphyrin and fullerene. So, you have the carotenoid, you could see the long stretch molecule, long tail molecule at A. Then you see the porphyrin, which is sitting out there, which is an molecule which is the same molecule as your chlorophyll, and you have the fullerene ball on the other side. Okay. So, there is a single step semiconductor particle attached hydrogen oxygen evolving co-catalyst. So, this is the oxygen evolving evolution catalyst and hydrogen evolution catalyst. So, these are the kind of geometries what are being followed. If you go to the 2 excitation process, where you have 2 different molecules. So, which you follow the C, you see the 2-step system, mixture of semiconductor particle with attached hydrogen oxygen evolving co-catalyst and redox electrolyte couple. So, this is very interesting out here, where you have 2 system which is basically trying to emulate this kind of system, where you see that one chromophore and the second chromophore mimicking the Z scheme of things. And then you have the electrode system. So, this whole area has evolved at 2 different levels. So, you need to develop high end electrodes for this, and you need to develop molecules which will split the water, as well as you need molecules which will by the action will absorb light and do this job. So, where this all started, you have to go back in time. So, this is the major breakthrough in 1972 by Honda and Fujishima, who actually during this was his PhD problem in Tokyo University while he was doing it. What he was doing, he was developing titanium dioxide for a totally different purpose, and what he found out is that pretty much laid the foundation stone of self-cleaning glasses. He was using, so now if you kind of go through the slide, who used a titanium dioxide film as a photoelectrode, but no co-catalyst on the titanium oxide surface. And this titanium oxide surface, the photo-generated electron was separated from the whole at titanium dioxide and electrolyte interface. So, this was the first evidence where titanium dioxide could split the water, the oxygen and hydrogen generated at a titanium dioxide electrode and a platinum counter electrode respectively in separate compartments inside the photoelectrochemical cell. So, this was the first, you can say that this was the first breakthrough which laid the foundation stone for photoelectrochemical cell. And what a photoelectrochemical cell is, I will give you the reference, but just for your understanding here, just for its simple understanding. So, it's basically the principle of operation of is based on the N-type semiconductors, and I will give you the exact reference. You please go through what is PECs. Basically, PECs are a hybrid device of semiconductor and electrolytes. Okay, these are not the pure dry kind of situation. So, there is an electrolyte and there is a semiconductor, and I will give you a very nice reference which I have already highlighted here by Michael Gritzel on the photoelectrochemical cell. That is the title of the paper. Please go through that paper. I really wish you people to go through that. I will be supplying the paper anyway. Go through that paper, because that paper will give you an idea about how this whole concept of photoelectrochemical cell, because that will be needed in order to understand the Gritzel cells or the dye sensitized solar cells. Okay. So, one second before I coming back to where I was. So, yeah. So, there are 2 levels where these kind of things have to be developed. The other level is the, so let us divide the problem. So, you need to develop the, let us enumerate it. You need the water splitting chemicals which will split the water. You need an electrode system which will capture the electrons, okay, and then you have to put this whole thing in one assembly. So, the last 2, DEF what you see is essentially the electrode system what has been developed. Single excitation step of water splitting cell containing a semiconductor electrode with water oxidation co-catalyst and a counter electrode to reduce water. So, you have both the things. On one electrode you are oxidizing the water, there is another electrode where you are reducing the water. Then you have dye-sensitized transparent metal oxide water splitting cell and the two-step tandem water splitting cell. So, these are some of the examples of it. Now, coming back to what are the new concept of nanomaterial development. So, if we follow this part 1, which is zinc oxide nanowire array electrode on the fluorine doped tin oxide transparent conducting substrate. So, you need different kind of transparent conducting substrate, because this process has to be light dependent. Light is the source of energy in order to carry out this process. Okay. So, there are different kind of fluorine doped tin oxide which are being developed over a period of time, which acts as a transparent conducting substrate. So, there is a lot of work which is kind of currently going on zinc oxide surfaces and developing different kind of material which are transparent enough to allow this process to take place. Okay. Please go through the paper reference what I have given you, because that will give you a better idea about what all development has happened in this area. And this is what will give you an idea of the photo-induced charge separation and recombination of a semiconductor photoanode. This is basically the basic very fundamental principle of the photoelectrochemical cell, where you have an interface of a semiconductor and a electrolyte, and where the water splitting is taking place. So, this is basically what is happening in a photoelectrochemical water splitting cell employing a photoactive anodic electrode that induces a space charge region at the semiconductor solution interface. The photogenerated electron is separated from the interface and then consumed to generate hydrogen at the counter electrode. A hole is generated at the interface and consumed for water oxidation reaction. So, this is pretty much what is happening. So, you are splitting the water, you are getting the protons through a membrane on one side, where you are again reducing it by adding electron to make hydrogen. Okay. So, this is the overall kind of scheme of things. And if someday, if this whole thing really works out, that we are able to mimic the manganese cluster in near future or in far future, these are some of the. So, if you look at this, so this is kind of a dream where we will have hydrogen power plants which will be present, which will be generating hydrogen, and hydrogen will be supplied as the major energy source for pretty much everything what you see all over the, apart from the other sources which are like photovoltaic power, underground and the wind power, and likewise and so on and so forth. So, what I wanted to highlight here is that you have to realize that biology is no more just a community we are studying all these molecules, but biology is an inspiration now, biomimicry, biomimetics. It is an inspiration for generations now of physicists, chemists, electronics, who are trying to emulate these wonderful structures what nature has already developed. Because even if we could understand, as I always tell, that 10% of it, we can really, not only we can dream, we can really achieve clean and green energy all over the earth. So, in that whole process, so as I told you in the beginning of the lecture, that we'll be talking about the two set of development. One set of development is in terms of the hydrogen fuel. The other set of development will be where the chlorophyll dye has been mimicked, or there are synthetic chlorophylls or the inorganic molecules like ruthenium and all, which is the ability to behave like chlorophyll. But those things always needed whenever we talk about natural, whenever we talk about those kind of dyes, they always needed a system, again that what I was trying to tell you, this one. You always needed a source of electron which will ensure that once they get oxidized by donating an electron, they should be brought back to their ground state. So, this is very, very essential for us to understand that this whole process, if you follow these 3 lines very carefully, who is the donor of electron, who is the acceptor of electron, and who is the perennial source of electron. It is all about these things, where this whole photosynthesis, water splitting, photosystem 2, photosystem 1, hydrogen generation, and pretty much all the power generation processes happening in biological systems are kind of governed. So, this is the overall scheme of things, and in next lecture we will be talking about the dye sensitized solar cells, followed by one of the lectures, probably the 38th lecture, we will be talking about iron disulfide solar cells and iron disulfide. So, these are the different inspiration which has been drawn over the period of time in order to emulate some of the feat which nature has already achieved. We are just trying to mimic nature. So, with this I will close in, and please go through all the references what I am giving, because they are really very good references. Please, because within the limits of a class you cannot explain everything. So, you have to really go through some of these references, wherever it is needed. I try to go as much as I can, but it is a generic audience, so I will have to be careful that, but please read through those references, because they will help you to clear some of your basics. Especially please read through this nature photoelectrochemical cell references. This will be very helpful to understand grid cell as well as the hydrogen generation. Okay, thanks a lot.